Okay, thank you for the invitation to come. I realise I'm standing between you and a cold drink, so I'll try and make this as quick as I can. I confess straight up that I left the word boron out of my title in my abstract, and so if you read the abstract you'd think that we had a problem with high soil in Australia, but in fact it's a problem with high soil boron, so let's get into it. Okay, so what I'm going to do in the next 20 minutes or so is just give you a bit of a background for those that haven't perhaps heard this story before. I'll talk a bit about our published work in barley, and then I'll go into some of our more recent work in wheat that we haven't published yet, and hopefully some of that will be novel to some of you. So this is what boron toxicity looks like. It presents itself as necrotic lesions in the leaves. Usually they appear in the tips of the oldest leaves first, and this is because boron accumulates in the, enters the plant, accumulates in the tips of the oldest leaves at the end of the transpiration stream. This is a map here of South Australia. We're based in the southern part of Australia. We're in Adelaide. The zoomed in area here is the bottom half of our state. The green and red regions represent our grain growing areas, and in red are the areas where boron toxicity is a problem, generally above 15 kilograms per kilogram in the soil, and that's more than about 70% of our grain growing regions where this is an issue. So we have marine soils of marine origin in Australia, and that's basically the reason why we have a big problem with boron. Seawater is high in boron. Soils are laid down as marine sediments, typically are high in boron. We have very shallow soils also in South Australia, and you can see here just going down through the soil profile we have a really large increase down to about 20 or 30 centimetres of soil boron in the profile. So if you drive north from Adelaide in spring, this is sort of the beautiful countryside you'll see, but then if you get down into this barley crop and have a look, quite often what you'll see are symptoms like this within the crop, and this was actually in a variety called Hindmarsh. I think this was taken in our last growing season in South Australia. So we have big issues even in currently released cultivars. So I think it's worth pointing out, for those that you aren't familiar with our environment, we have quite a different environment through the southern part of our country as opposed to the north. So in the south we really have predominantly winter rainfalls compared to the northern growing regions where there's generally higher rainfalls throughout the summer months. And this actually has a big difference on the levels of boron in our soil. So toxicity is a problem through our southern environments, whereas if you look in the north, it's actually more to do with boron deficiencies that actually is the challenge in those environments. So there's a large study done recently looking at about 230 trials over about 12 years by Glenn MacDonald in Australia, and it showed on average that tolerant genotypes have about a 16% yield advantage over intolerant genotypes in southern environments of Australia. So in both barley and in wheat, boron toxicity is associated with two main phenotypes. The first is leaf symptom expression, and this is to do with boron accumulation in the leaves, but also it's to do with root stunting, and you can see here quite clearly that boron intolerant lines have a very stunted root growth compared to boron tolerant lines when both grown under high boron conditions. So that's the basis of the phenotype that we've been using to do all of our positional cloning and mapping work in barley and in wheat. So just quickly, in barley there is great genetic variation for this trait, and that's been the basis for all the mapping work that we've done. Tolerance essentially in barley is derived from the basis of tolerant genotypes to efflux boron from the roots. So you see that tolerant genotypes grown over a range of boron concentrations, this is in hydroponics, have the ability to maintain a much lower leaf boron concentration than do intolerant genotypes. And quite clearly you can see here the difference between tolerant genotypes and intolerant genotypes when applied with high boron. So in barley, for more than 30 years actually, this has been work undertaken in Adelaide, and this is work done by Steve Jeffries in the late 90s. This was work mapping in a doubled haploid population derived from a South Australian malting variety called Clipper and an Algerian landrace barley called Sahara 3771. There were two major QTL identified on chromosome 4 and 6, also two other minor ones on 2H and 3H. So we targeted both of these major QTL in early work in this program. This is some back-crossing work. Now I'm not sure how clear this comes out, but this on the left here is a So these are both some Barley back-cross lines. On the left are the back-cross lines that are missing. Most of the, or I think it's 3 out of 4 of the major tolerance QTL, and on the right here is a similar set of lines containing the QTL, and I hope you can see the difference in leaf symptoms there. So we used a pretty basic approach. I'm not going to go through this. I think this has been described and probably a fairly familiar concept to most of you, but we start with a trait or locus. We use a fairly broad genetic map. In barley or wheat we use synteny to rice or praecarbodium to saturate this region with markers. We end up doing fine mapping, and then through use of back clones we hopefully end up with a candidate gene through the locus. So that's been our approach in both barley and in wheat. So in barley we cloned a gene at the 4H locus. It's a boron efflux transporter. It's actually an anion permeable channel. The basis of the allelic difference between Clipper and Sahara here is mainly that Sahara has a tandem duplication of genes at this locus. So there are at least 4 copies quite closely tandemly duplicated at this locus, compared to intolerant genotypes that generally have 1 copy at the region. This results in a really high level of expression in the tolerant genotypes. And one of the interesting things that we noted early on when we were looking at the functional characterization of this gene was that it's expressed very highly both in roots and in leaves, and we noticed a very high level of expression at the very tips of leaf blades in barley. And we thought that this could be involved in perhaps the exudation of boron from the hydathode, which is the structure at the tips of the leaves, which results in this guttation of droplets that you quite often see under high humidity conditions. And it turns out that if you go back to the literature, you actually find some quite interesting work that showed that these guttation droplets have very high concentrations of boron. This is work from the early 60s. If you put a boron-tolerant genotype into a hydroponic solution containing boron, very rapidly the boron concentration in that droplet at the end of the leaf is higher in concentration in boron than the concentration of the solution that's going. And so there's some process there that could be involved in the removal of boron through that mechanism through the leaves as well. Okay, so we also identified a gene on a chromosome 6. And I'm not going to go into much detail about this. This is a similar approach using a map-based cloning effort. This is an influx transporter. It's part of the aquaporin family of transporters. It's actually an ortholog of the ricilicin transporter LSI1. In this case, the Clipper and Sahara proteins are identical. The basis for the allelic difference here is based on reduced expression in the tolerant genotypes, so there's less boron getting into the plant. So this is what we think is going on in barley. This is a schematic showing a root of a tolerant genotype and a sensitive genotype. So the purple arrows show the passive movement of boron that happens in both genotypes through the plant cell walls and through the membranes. In tolerant genotypes, we have a relatively lower expression of the aquaporin gene, which results in a fairly small amount of boron coming through. Tolerant genotypes have a much higher expression of the efflux transporter, again resulting in smaller amounts of boron coming into the xylem compared to intolerant genotypes. And the net effect of that is that tolerant genotypes generally have a lower level of boron getting up into the shoots. So moving into wheat, so there is great variation for tolerance in wheat as well, and this is also work that's been going on for perhaps more than 30 years at the Wake campus of the university. A lot of the initial mapping was done in 2 lines, a tolerant genotype by the name of, a tolerant genotype by the name of Halberd, and an intolerant genotype called Cranbrook. So again, early genetic work showed that there's actually one major QTL on chromosome 7b. This is controlling around 65% of the phenotypic variation for tolerance in this trait. So a major QTL, and that was the region that we targeted. So Torsten Schnurbusch was working with us many years ago, and he started a lot of this work. Margie Plotter has been continuing that after he's left. So again, I'm not going to go to the details here, but I just wanted to show this to show that we used a syntenic approach using rice brachydipodium to try and saturate the region around BO1 using fine mapping. I'll come back to this at the end, but it's a complex region on chromosome 7. There's a lot of things going on. There's a lot of different genes and loci involved. So that all sounds simple, but that actually took around 10 years to find map that gene, and there are a few challenges that we sort of hit along the way that I think sort of illustrate the problems that this approach can have. And the first was that we noted a fairly low level of recombination in this region in Cranbrook and Halberd. The gene is not present in rice. The gene is not present in Brachydipodium. At that stage, there weren't any gene sequences for the tolerance gene present in any sequence databases. And also the gene is not present in the back library that we initially started screening in Langdon, Durham. So we eventually came across the gene after we started screening a Tauschi back library, and this was in collaboration with the work of Johan Dvorak. So it was a challenging process, but we got there in the end and we cloned a transporter gene. It's similar to the Barley transporter that we'd cloned on chromosome 4, but it's important to note that it's actually not the direct ortholog of the Barley gene. So we developed a mutated population in the tolerant genotype Halberd. We found several EMS mutants in the gene. One of them I'm just showing you here. This is a mutation in one of the exons, causes an alanine to valine change in the amino acid sequence. You can see here that under low boron conditions there's no difference in root growth between the tolerant genotype Halberd or homozygous lines, homozygous segregants for the wild type phenotype or homozygous segregants for the mutant. Of course, when you hit them with high boron, you clearly see that there's a stunted root growth. There's been a reduction in function of the transporter. We've done heterologous expression for this gene, and you can see that when you make this change, you reduce the growth of yeast expressing the transporter. They're effluxing less boron there, therefore they're not growing as well. So we're interested, this is very much a breeding focused project, and we're interested in looking at allelic variation to try and support the breeding programs to improve the breeding efficiency for this trait. So we started looking at the allelic diversity through wheat germplasm in Australia. So this was a big job, and what we found was really quite interesting. So we found a big spectrum of allelic variation for this gene. So I've just sort of tried to illustrate these schematically here. So if we start at the top, the red schematic here is the tolerant allele from Halberd. Okay, so this is located on chromosome 7b. We've also identified a duplicated version of this gene on chromosome 4a. I'll come back to that later. But we also see a range of different allelic forms that range from intolerant through to completely non-functional forms. So there are lines, for example, like Chinese spring that contain intolerant versions, partially functional versions of the allele, and we believe this is due to insertions in the promoter sequence that drastically reduce the function of the transporter, the expression I should say. There are also intolerant alleles from different cultivars that have deletions within the gene that cause non-functional versions. There are cultivars that have highly truncated versions, and there is our intolerant parent of our mapping population, Cranbrook, which has an actual full gene deletion. It's a null at the locus. So a broad spectrum of allelic variation for this gene. Copies on 7b and 7d, but nothing on 7a. So again, we looked at the phenotype of plants containing those different alleles. Remember that the tolerance versions are here coloured in red. The intolerant alleles are these other colours here. So if you look at root length growth of cultivars containing these different allele types, you clearly see that cultivars containing the tolerance allele maintain their root growth under high boron conditions, whereas those that don't have that stunted phenotype. And generally speaking, tolerant genotypes, those coloured in red, have a much higher expression of this transporter than those with the non-functional or much lowly expressed versions of the gene. So there is actually a rare allele that we found on chromosome 4a. It's a rare allele. It's absent in most cultivars that we've looked at, and A genome progenitors of wheat. We found it in a landrace wheat from the Mediterranean. This is a line called Greek, or G61450. So it's a very rare event. It has very high expression. You can see here the very high expression of this transporter compared to the Halberd tolerance gene on 7b. It's almost identical. It has one synonymous SNP with the halberd gene. We don't see any evidence of a translocation to this region 4A, so we think this is a dispersed duplication event of this gene from 7B to 4A. So we're also interested, as well as characterising the different forms of these alleles, we're also interested in looking at the allelic distribution across the Australian environments. And remember I said before that it's mainly our southern environments that have problems with boron toxicity, and it's actually the northern environments, coloured in blue there, that have the opposite effect, and that is usually with low boron conditions. So we're interested in looking at the allele frequency amongst lines that were bred for those different environments. So we assembled a collection of advanced breeding lines and released cultivars from one of the major breeding programs in Australia, Australian Grain Technologies, and this was a mix of southern material and northern material. And what we found was quite interesting. So we looked at the allele frequency in lines that were bred for the southern environments, and we compared that to the material developed for the northern regions. And what we saw was a dramatic difference in the allele frequency of the tolerance gene in southern environments compared to the northern environments. And in fact, this one line that was bred in the northern region was actually bred in the northern region but targeted to the southern environment. There are actually no examples of wheat lines bred for the northern low boron regions in Australia that contain the tolerance locus. So there's obviously been indirect selection for the tolerance allele in these areas, and this may seem obvious, but in fact it's information that was new. The breeders didn't know this. And what we see now is also that tolerant genotypes are always inefficient under low boron conditions. So we also are interested in looking at the origins of boron tolerance in wheat in Australia. We're interested in knowing where this came from. So we selected a set of wheats from around the world, and we tried to sort of ask the question about where this tolerance locus came from. So here I've just shown in orange we have countries that are coloured that have some evidence where boron toxicity has been documented in the literature or anecdotally. The black diamonds here show the hypothesised source of tolerance alleles that we think is the location of where they came from. The arrows show the dispersion that we think has happened, and then the countries with the red dots here are the ones where the tolerance alleles we know are identified today. So we're able to follow this dispersion out of the Mediterranean. We know now that the boron tolerance that we see in tolerant genotypes in Australia and South America appears to be of tetraploid origin, and we think we can track this through to a landrace barley called cretin that was used to develop the 20th century wheat line Currow. It was actually a parent of the current tolerant genotype in Australia called halberd. So there's been conservation of this tetraploid diet-derived segment in modern wheats in Australia, and we're wondering whether this has presented a barrier to recombination and might present issues for utilisation of other linked loci that are tightly linked to this boron tolerance gene in wheat. And one of those is late maturity alpha-amylase, and I'll quickly give you a little bit of a rundown on why I think that might be an issue. So this region on the long arm of chromosome 7 is a busy region. There's a lot going on. One way to describe this is a bit of a street party. Everyone's welcome. There's quite a lot of resistance loci here. One of the loci that's very important to breeders and to growers in Australia is called LMA, for late maturity alpha-amylase, and I'll tell you a little bit about that. So this is a genetic defect in Australian wheat germplasm. It is the production of alpha-amylase midway through grain development, and it's a serious quality defect in Australian wheat. So what happens is that, often triggered by a temperature shock, you get the production of alpha-amylase before it really should be happening around maturity and germination. And then the important concept here to get is that the halberd 7BL source of boron tolerance is actually the desirable form of LMA, so they're tightly linked together. So you get halberd boron tolerance and you get the desirable form of LMA. The phenotype is very difficult for this trait. It's slow. There's no visible difference in the grains between LMA and non-LMA types. It doesn't affect all the grains in a spike, and it's severely affected by RHT genes which interfere with the trait expression as well. So it's a rare event, this non-LMA locus. It's for non-LMA and halberd, and this is desirable by growers in all environments in Australia. The halberd 7BL boron-tolerant source is, of course, as I've described to you, is desirable in southern environments, but it's an undesirable characteristic in the northern environments. So what's happening is that growers that are looking, or breeders that are looking for the source of non-LMA from this rare source in halberd are also getting the tolerance locus for boron in halberd. And we think that a suppression of recombination through this region because of the tetraploid source of this locus may be one of the barriers to effective breeding and separation of these two traits in wheat breeding. So one of the outcomes of our sort of map-based cloning effort was that we identified a rare recombinant between tolerance and non-LMA, and so we've been able to give this to the breeding programs to improve breeding for both of these traits. So we're having a bit of a positional cloning approach to this to try and clone the gene responsible for this LMA phenotype, and I won't go through the details here, but just to you follow the idea here. We're currently looking at the genes in this interval here, and we're using a minimum tiling path of bats through collaboration with the Norwegian group, looking at sequencing of chromosome 7b through the Wheat Genome Sequencing Consortium. So the differences between wheat and barley, I think, are quite interesting. I think we have evidence to show that tolerance in wheat and barley arose through the divergent evolution of paralogous genes. There's transcript level variation, which is a common determinant in wheat and barley. But what we know is that the tissue specificity and the inducibility of the tolerance genes in barley and wheat differ. And in wheat, the important thing is that there's very broad, or comparatively broad compared to barley, allelic variation. And this has provided adaptation to wheat in diverse environments. So we think the wheat tolerance locus is of recent origin, definitely post-domestication. There's been a big impact of selection by early farmers selecting land races. We know there's been a big impact of duplications in adaptation in land races to boron tolerance. And there's been a high level of importance in these land races to the development of locally adapted boron-tolerant varieties in Australia. And there's been strong allelic selection. So what we know now from our work looking at allelic variation across Australia is that matching alleles to environments is certainly critical. So that's all I wanted to say. I just wrap up by thanking the people that have done most of this work, and most recently that's been Margie Pilotta in our lab, who's driven a lot of the weak positional cloning work, Julie Hayes, also Alison Hay, and prior to that, Torsten Schnurbusch. So I thank you for your attention. I'd be happy to answer any questions. Thank you. That's okay, no problem.
So I've got two questions. Number one is, what is the trade-off of this boron tolerance? That means, why do farmers not want to have the tolerance if they grow wheat or barley on non-boron soil? And the second question is, how specific actually are those importers or exporters or transporters? How specific are they for boron?
Yeah. Okay. So the first question, well, in barley at least, it's a constitutive tolerance mechanism. So the protein is always there, effluxing boron from the roots. So in low boron environments, what actually happens is that tolerant genotypes suffer from boron deficiency. So they're effluxing so much boron from their roots, either out of the roots or to the apoplast, that they don't get enough up into the roots and into the floral structures where it's actually required for pollination. So that's the major reason, is that it's deleterious in those environments because it's probably imparting a deficiency phenotype. And so the specificity, we're trying to address that question now. We have a couple of PhD students in Adelaide working on the protein aspects of those two genes from barley and wheat. Actually, they're protein structure function projects. The the efflux transporters, the 4H gene and the 7B gene in wheat, we think are very specific to boric acid. In fact, they're probably transporting the borate anion. The 6H locus in barley is a transporter of metalloids, so it transports boron as well as germanium, silicon, arsenic. So we think the major loci are very specific to boron. Yeah.
In wheat, you said that boron tolerance has origins from tetraploid wheat. Is that durum that you're referring to? Is it the progenitors of durum wheat?
Yes, that's correct. Yep. Yep.
More questions? Do you have enough? Okay.
Just to comment, positive of course, because first of all you showed the importance of durum, right?
Thank you. I thought you'd like that, Roberta.
Take it as a joke, but I think it also shows that once you start cloning these elephants in the room, like I like to call them, you can switch back and forth with the different allied species and gene pools, and we learn a lot about how these genes function. Out of curiosity, have you investigated to what extent the increase in expression might be due to a long-range enhancer, or could be something related to the promoter of the... Have you investigated that?
You're talking about in barley, where there's a tandem duplication, or are you talking
No,
in wheat?
I'm talking about also wheat. You have a different expression.
No, we haven't. We haven't done any transgenic work to try and look at the promoters of those genes.
But if you do the fine mapping, the QTL peak is smacked on the structural gene, or it's upstream?
Well, the resolution of the QTL mapping is not that good.
Okay, because I'm thinking,
So
with
we don't-
BGT1, we found we have a similar situation, but the association mapping brought us 70 kb upstream, so I was curious.
Yeah, we haven't got the resolution to do
Okay.
that, to answer that question.
Thank
but
you.
it'd be...
But do you have SNPs in these open reading frames, in the two parental alleles?
Well, in Halberd, in Cranbrook is a null. It doesn't have the tolerance.
Ah, it's
gene.
a null.
It's
Okay.
completely absent.
Ah,
The locus is just not there. So
Sorry, I missed that.
there's one big snip.
Okay, yes, that's a big thing. Sorry, I missed that.
Yeah.
Okay, thank you very much, Dr.
Okay.
Sutton.
Thank you.
We have enough time for previous two speakers.
Okay. Roberto, I have a question for you. I'm very interested in your peduncle length QTL. I'm wondering, have you had a chance to look and see if it's in any way related to the ASI QTL in maize for drought tolerance, which is also basically the equivalent of feeding the peduncle?
Okay, let's put it this way. First thing we thought was, and then I have a question for Dr. Prasanna lately, because the reason I like to clone QTL is because I like to understand what's behind from a physiological standpoint. So my first thought, I bet it's going to be water status and roots. Nothing. Basically, what we find a physiological standpoint, it's different in stay green that is also related to soluble, water-soluble carbohydrate. We have not looked at what you have said, you have asked. I think my impression, which I bring in the loop also Dr. Prasanna, is that ASI, it's a nice, quote-unquote, spy of the water status of the plant, which in a way it's very intriguing, but again, to me, it's much more interested question for Dr. Prasanna. What is behind these QTLs? Is it flowering time? Is it escape? Is it avoidance? Is it fruits? Because as a breeder, I would be much more intrigued in going that direction rather than simply saying, okay, it is a proxy. Yeah, it is a proxy, so it is useful. So I strongly doubt that there is anything due to the fact that, again, we have tested the water status, we have tested the roots, and we don't find any evidence whatsoever. So now I'm off the hook, and I got Dr. Prasanna in the loop.
Can ask.
No, I'm just kidding. Yes, this is a very important question. What lies behind traits like ASI? Are the genomic regions, are there any functional candidate genes that associate with this. This is something which right now Zaidi is trying to investigate through the rhizotronics facilities which has been created in the ICRISAT campus, looking at what is efficient water use in different lines that capture different PSI variation, and how the structural and functional root rates influence the responses of specific CIMID donor lines against water stress deficit. So this is a progress, a study in progress. Now it cannot be undertaken under general water conditions or under stress conditions in the field. However, these are not escapes. CIMID's major focus is on developing drought-tolerant germplasm under medium maturity and full season maturity. So therefore, and flowering time differences are accurately captured and accounted for in all our statistical designs. So it's only in cases of extra early maturity or early maturity, you may say that ASI could be linked with the drought escape. But I genuinely believe that in medium maturity and full season maturity, and with the kind of rigorous managed stress screening protocol we have, there is no question of only escapes contributing to tolerance. Yes, absolutely. I was surprised when Babu has found out not only DREP1, DREP2, lipoxygenase, ABA-related genes in some of the major CIMID drought donors. So it could be related to some of the genes you are interested in probing in maize.
Last question, was the high ABA associated to high yield or low yield?
Not yet. That is not yet analyzed. So okay. This is a recent study which he did it on back-cross derived DH lines.
I bet a beer that is associated to low yield.
Okay.
But I'll lose my bet.
Okay.
Following up on the same interaction with the two folks, I'll bet the opposite way. Why? Because in pearl millet, our drought tolerance QTL, which is at the same position as your chromosome 1L drought tolerance QTL, is a constitutive elevated ABA that causes partial closure of the stomates, reduces water consumption or water use during vegetative development, leaves something down there to be used during grain filling, provided your soil is heavy enough that it doesn't just percolate on through.
Whoever wins has to give wine to all people.
Yeah. Yes, at your meeting next year, no problem. Very dependent upon the soil.
Well, anyhow, thank you very much for all the three speakers. Let's clap again for them.
Thanks everybody, and on behalf of organizers, I would like to thank Dr. Tuteza and Dr. Balyan for having such a great session, and I would like to thank Dr. Tibrosa, Dr. Prasanna and Dr. Sutton for giving such a good talk, and I hope that definitely, and even I believe, this will help us to further work to improve our crops. Thank you so much.